So, dear everyone, welcome to this week's episode of the ESDR Kitchen, which will be a freshly baked with two outstanding speakers. We'll just wait a few seconds until everyone joins from unregular or unexpected guests here. Let's put it that way. The usual hosts, Kilian Heinrich and Sondin Drübach, are on holidays or unavailable. So Enikö, Sonkoly, and I have the pleasure to actually host this week's webinar, which is sponsored by BMS, so with glad support from Mr. Maya Squibbs. And today, as mentioned, is a freshly baked, so we really have two outstanding publications that have been recently published this year. They were chosen by Kilian and Sondrin, but again, we just have the pleasure to actually be here. So I'll just start with an introduction of the first speaker, which is Piotrek Konieczny. For me, it's still a bit difficult, even if he's a veteran and also second time that he's actually presenting here. I'm sure he'll excuse me for my not so perfect pronunciation of his name. So Piotr is currently K99 National Institute of Health Fellow at the Icahn School of Medicine in Mount Sinai in New York. In 2019, after getting his PhD in Poland, he joined Curdin's lab and did a postdoctoral there. He had some absolutely outstanding publication, including Science, etc. And now just recently again in immunity, and he's going to present this today. Thanks for joining. He also received some research for his, as he searched for some awards for his research. And probably the most important one is the actually research award for a young researcher that he received from the ESDR. So, Piotr, thanks for coming. I'll keep it short. The stage is yours.
Thank you so much, Conrad, for this kind introduction. And hello, everyone. I will share my screen. Can you see everything?
Perfect.
Okay. Okay. Hello, everyone. Thank you so much for invitation and for the opportunity to discuss our recent published story, which really center around this concept around this crosstalk between two different skin compartments, immune compartment and skin epithelium compartment, and how this crosstalk drive metabolic codependency in skin inflammatory disease, in particular in psoriasis. Okay, so as you know, every single day, our skin as a barrier tissue is exposed to different environmental inputs, different types of damage. For example, like when we have a mechanical damage, like acute injury or inflammation damage, where our immune system is hyperactive and cause skin pathology or infection damage. And these different types of environmental inputs or different types of damage are sensed by homeostatic immune cells that are present in the skin, and these homeostatic immune cells engage with the crosstalk with other skin cells, and this crosstalk is what really mediates this tissue adaptation, skin adaptation to environment. And we have previously described this adaptation in the context of skin repair in skin acute injury, where we found that homeostatic lymphocytes, the subset of homeostatic lymphocytes, which we call our gamma T, gamma delta T cells, they rapidly respond to skin injury, and they secrete interleukin-17 that is directly sensed by damaged epithelium. And interleukin-17 was required for HIF-1 alpha activation, so for activation of hypoxia-inducible transcription factor, which drive glycolysis metabolism in skin epithelium, which was required for optimal skin epithelium revitalization. So we really wonder whether the same mechanism that drives skin repair can be co-opted in skin pathology, where we observe this damage repair failure, more specifically in the context of skin maladaptation in inflammatory skin diseases, psoriasis, which I will discuss today. With collaboration with two fantastic scientists in the lab, Dr. Ipsita Subudi and very talented bioinformatician, Alexandre Prustyba, we really ask a very simple question: is this axis, which involved interleukin-17 and HIF-1 alpha signaling, is co-opted in inflammatory skin diseases, more specifically in psoriasis? I think I don't have to introduce psoriasis to this audience, but the main reason why we decided to focus on psoriasis is because in psoriasis we observe this unchecked immune epithelial circuit, which is driven by type 17 immunity, type 17 immune cells that secrete interleukin-17 that cause this keratinocytes hyperactivation and inflammatory mediators secretion. So first, we really wonder whether HIF-1 alpha is present in psoriasis, in psoriatic lesions, and if so, where. So first, we took two different approaches. The first approach, we compare HIF-1 alpha expression together with interleukin-17 receptor expression, more specifically interleukin-17 RC receptor in lesional skin, so with active psoriatic plaque and with non-lesional skin, and we found that HIF-1 alpha is highly expressed in skin epithelium, in lesional skin, in the cells that highly express interleukin-17 receptor. And the second approach, we look at HIF-1 alpha gene expression as well interleukin-17 receptors expression in the patient that were treated with anti-IL-17 blocker with secukinumab. And we compare patient samples before and after treatment, and we found similar results that HIF-1 alpha is highly expressed in the patient in lesional samples before treatment with anti-IL-17 blocker, and after post-treatment we observed this significant inhibition in HIF-1 alpha expression as well interleukin-17, which really suggests first that this axis that involved interleukin-17 and HIF-1 alpha is present in psoriatic pathology. As well, it suggests that HIF-1 alpha is a downstream effector of interleukin-17 signaling. So to test the functional contribution of HIF-1 alpha as an effector of interleukin-17 signaling, we perform a small clinical trial on the dish. This is one of my favorite experiments, where we took human skin punch biopsies, either from healthy patient or patient with psoriasis, and we cultured them ex vivo on the dish for 24 hours, either with vehicle control or standard of care, so with corticosteroids or with HIF-1 alpha inhibitor. And after 24 hours, we isolate RNA and we perform RNA sequencing. And here I'm just showing you the volcano plot with the genes that depends, the expression depends on HIF-1 alpha signaling. These are downregulated genes in the psoriatic sample that were treated with HIF-1 alpha inhibitors versus vehicle control. And we project this genes, the signature that depends on HIF-1 alpha signaling, on publicly available clinical trial with anti-IL-17 blocker with secukinumab. And we found we were very excited and surprised to see that that HIF-1 alpha signature significantly distinguished anti-IL-17 responsiveness from placebo group, as well as stratified therapy responsive versus non-responsive patient. And this really suggests that that HIF-1 alpha transcriptional program is important driver of psoriatic pathology downstream of interleukin-17 signaling. So of course, I think the biggest question was what's the function and role of HIF-1 alpha in psoriatic in psoriasis, and so we turn on to our favorite mouse model, Enikö model. So it's a model of psoriatic-like model in mouse. So we generated epithelial HIF-1 alpha deficient mice. We treated them with Enikö, and we found that by inhibiting HIF-1 alpha specifically in skin epithelium, we almost completely block psoriasis development, which we measure based on keratinocytes hyperactivation like epidermal thickening or hyperproliferation. But what was more important for us, we not only observed the defect that by blocking HIF-1 alpha, we block keratinocytes hyperactivation, but also we observed that by blocking HIF-1 alpha, we also blocked immune infiltration, more specifically gamma delta T cells, as well other features of psoriasis like hypervascularization and hyperinnervation. We really suggest that HIF-1 alpha is an inflammatory factor in the specific context that drive skin pathology. And to make this story short, this long story short, the downstream of interleukin-17 and HIF-1 alpha signaling was metabolism, was glycolysis metabolites, where we found that genes that are the expression of the genes that are involved in glycolysis are dependent on HIF-1 alpha activation and HIF-1 alpha signaling. So we generate glucose transporter 1 deficient mice, specifically in skin epithelium, and we treated mice with imiquimod. And again, we found that by blocking epithelial glycolysis, we also block psoriasis development, keratinocytes hyperactivation, and what was more important, we blocked gamma delta T cells infiltration as well. We observed that the cells, the gamma delta T cells, they significantly secrete less interleukin-17 compared to wild type mice, which really suggests that this epithelial glycolytic shift, which is happening in skin epithelium, not only control keratinocytes skin epithelium pathology, but also control and drive type 17 immune response. So, you know, we really wanted to know which mediator, which inflammatory mediator downstream of HIF-1 alpha glycolysis drive type 17 immune response. So we spent a lot of time to look at very canonical, classical and canonical factors that drive type 17 immunity, and we couldn't find anything. So we took a step back and we focused on metabolism, and we found this paper from 2016 where they shown that lactate as an end product of glycolysis, a byproduct of glycolysis, is highly upregulated in psoriatic patients. So we confirmed that lactate is present, is secreted in psoriatic mouse model, where we took mouse skin punch biopsies, either from control mice or mice treated with imiquimod, and we cultured them for 24 hours ex vivo on the dish, and we found that lactate is significantly is highly secreted in skin samples treated with imiquimod, and this secretion was dependent on glycolysis. When we block glycolysis using 2DG inhibitor, we also block lactate secretion. So to confirm the contribution and function of lactate in psoriasis, we took three different approaches. We inhibited lactate production by using LDH inhibitor, which we topically applied on the back skin of the mice. As well, we blocked lactate transport by blocking lactate transporters MCT1 and MCT4. And again, we found that by blocking lactate, we not only block skin epithelium pathology by blocking keratinocytes hyperactivation, but also we blocked gamma delta T cells infiltration. And what was more important, we also observed by blocking lactate, we also inhibit interleukin-17 secretion by gamma delta T cells. And the last piece of the data, which I want to share with you, I think the biggest question and challenge was to understand whether lactate is sufficient and directly regulate interleukin-17 secretion by gamma delta T cells. So we isolate gamma delta T cells and we culture them with or without lactate, and we found that indeed lactate was sufficient to induce interleukin-17 secretion by gamma delta T cells. So this really brings us to this model which we present in the paper, where we found this pathological circuit between immune compartment, skin immune compartment and skin epithelium, where gamma delta T cells, they actively secrete interleukin-17 that it's sensed by damaged skin epithelium and induce HIF-1 alpha activation that regulate glycolysis metabolism and end product of glycolysis lactate, drive this loop and act as inflammatory mediators on gamma delta T cells and maintain and sustain interleukin-17 secretion, which was not only important from the perspective of skin epithelium from the keratinocytes, but other typical features of psoriatic pathology like hypervascularization and hyperinnervation. And with that, I would like to thank all NAIC lab members, especially Shruti, Ipsida, and Alex, my funding source, collaborators, and you for your attention. And again, for the invitation, I will be happy to take any question later. Thank you.
Thank you very much for this beautiful presentation of this work. It's indeed very nice, and I hope that the audience will have many questions. I think so. So please put your questions in the chat or in the Q&A, and we will take those after the second presentation. And with that, now it is my pleasure to introduce our next speaker today, who is Camille Braun from Lyon, France. Unfortunately, we cannot see him because he has some issues with his camera, but he can share his slides and we will be able to listen to his presentation. So Camille Braun is an allergist and pediatrician at the Hospice Civil de Lyon in Lyon, France, and a researcher in immunology at the International Research Center of Infectology in Lyon. And he has just completed a 2-year clinical fellowship in Montreal. So topic-wise, we will still stay in skin inflammation, but now we will focus on atopic dermatitis and Staphylococcus aureus, which I think is the question that interests many of us. What is really the role of Staphylococcus aureus in AD? So please, Camille, the stage is yours.
Thank you very much. You see my screen, yes?
Yes.
Yes, good. Thank you for this introduction. Thank you for the invitation, and sorry for the webcam. I'm very, very happy to present my work on the contribution of Staphylococcus aureus to the pathophysiology of atopic dermatitis and notably Staph aureus-specific skin-resident memory T cells. This work has been done during my PhD at the International Center for Infectiology Research in Lyon, under the supervision of Marco Faucanson and Audrey Nussbaum. I guess the first question you have to ask yourself is why this guy, an allergist and pediatrician, is interested by Staph aureus. In fact, patients suffering of atopic dermatitis are frequently colonized by Staph aureus, even before the onset of the disease. And this colonization is frequently correlates with the disease severity and correlates also with food allergies, suggesting a role of Staph aureus in the disease pathophysiology. As you may know, atopic dermatitis is characterized by a skin barrier defect and a mixed inflammation with markers of type 2 immunity, but also type 17 and others. Both innate and adaptive immune cells, mainly lymphocytes, are thought to be involved in such inflammation with many different triggers, including irritant and allergens. Despite several studies have shown that Staph aureus produce several pro-inflammatory factors, notably virulence factors, the role of Staph aureus colonization in the disease course is unknown. And to clarify it was the aim of our study. We notably had 2 hypotheses that could participate to the initiation of atopic dermatitis and could also trigger the exacerbation of the disease or aggravate the inflammation. To answer this question, we used an original model by collecting clinical strains from patients with severe atopic dermatitis, and these strains were separately cultured and suspended to be painted on the ears of Black 6 mice with a swab. Importantly, the skin was initially intact, and we were able to analyze the primary response to Staph aureus, which corresponded to the inflammation induced on the naive skin, and the memory response after the resolution of the primary response, and eventually after the reapplication of the bacterium. The clinical inflammation was followed by measuring the ear thickness. So in this presentation, I will focus on the memory response, which provided many original data and obviously many questions for further studies. But to understand our model, I will briefly show you some data on the primary response. Firstly, the application of clinical strains of Staph aureus induced clinical inflammation on an intact skin. And interestingly, the intensity of this inflammation depended on the strain, with one strain called AD04E17 inducing the strongest inflammation, as you can see here, despite a similar colonization between the different strains. These strains, E17, will be used in the following experiments. When we characterized the inflammation by RT-qPCR, we found a molecular profile typical of mouse models of atopic dermatitis with a mixed immune response with type 2 inflammation, type 17, and also markers of type 2 inflammation, which is not usual after bacterial exposure. And we also found a polymorphic cell infiltration of both the epidermis and the dermis, including innate cells, monocytes, macrophages, innate lymphoid cells, as well as T cells, mainly dendritic epidermal T cells and dermal CD4 positive T cells. So all these experiments demonstrated that Staph aureus had the ability to induce an atopic dermatitis-like inflammation and could initiate the eczema. But now let's see what happens several weeks after the first cutaneous exposure to Staph aureus. At day 60, the inflammation was completely resolved. In the skin, by comparing a naive and healed skin by flow cytometry, we found the persistence of CD69, CD103 cells, which corresponded to resident memory T cells, mainly CD4 positive T cells and gamma delta T cells in the dermis. To assess the specificity to Staph aureus of these cells, we applied the bacteria on NUR77 GFP mice. In the cells, GFP positive cells are TCR activated cells. In this mice, after reapplication of Staph aureus, a significant part of resident memory T cells in green were specific to the bacteria, as demonstrated by the persistence of GFP positive T cells, alpha beta T cells, and gamma delta T cells. So in the skin, as you may know, probably resident memory T cells are responsible for hypersensitivity response, which is defined by an exaggerated inflammation upon the re-exposure to an environmental factor. In this condition, we call them an allergen. Here we have staph-specific resident memory T cells. Obviously, the next question is, if we reapply Staph aureus, will the inflammation be exaggerated as seen in hypersensitivity response? So we then reapply Staph aureus once and followed the inflammation for 7 days. And effectively, we obtained an increased inflammation compared to the inflammation after one application on a naive skin. And such inflammation was increased with higher concentration of the bacteria. We obtained similar response after injection of FTY72 suggesting that this response was due to resident cells and not circulating cells. And in model of depletion of T cells, so we depleted T cells just before the re-exposure to the bacteria, we abrogated this hypersensitivity response. Interestingly, both gamma delta T cells and CD4 positive T cells probably had a role in this hypersensitivity response. Last interesting data comes from the bacterial count after exposure to healed skin in comparison with bacterial exposure in naive skin. First, comparison of PBS conditions, so we didn't depleted T cells. We showed that hypersensitivity response was associated with a protection against bacterial persistence at day 7. And the depletion of T cells showed that this protection was provided by both CD4 positive T cells and gamma delta T cells. So taken together, all this data confirm that Staph aureus may have a role in atopic dermatitis pathophysiology. For the first time, we determined that this bacterium may have the ability to promote atopic dermatitis inflammation on an intact skin, but also to induce specific memory T cells that will promote an hypersensitivity response upon bacterial re-exposure. This memory response will also protect against bacterial persistence, and this data paved the way for further studies to understand how in patients with atopic dermatitis, staph-specific resident memory T cells may lose their ability to protect against bacterial colonization because patients have higher concentration of bacteria and are not protected against bacterial colonization, and how these resident memory T cells may interact with other cells to promote allergies, such as food allergies, since there is a correlation between Staph aureus colonization and severity of food allergies, for instance. So I would like to thank all my team and all the collaborators who were involved in this work, and I thank you for your attention.
Thanks a lot for these two excellent presentations. We'll go over to the Q&A session, as Enikö just said, and also written in the chat. If you have any questions, just write it into the chat or the Q&A, and we'll show them to or ask the speakers. We have a first question from Patra Vijay Kumar. Actually, he has two, one for Piotr, one for Camille. I'll start with Piotr. Thanks for the nice presentation. Did you see any other cells apart from gamma delta T cells producing IL-17, such as mast cells, when you interfere with lactate? Because lactate effect is not very specific for T cells and has a broad effect.
Yes, so thank you so much for this question. We have not looked much more broad. We mostly focus on type 17 immunity. So we have not looked on mast cells, and we know that gamma delta are the dominant source of interleukin 17 in psoriatic model. But I think like it would be very interesting to really look how broad this concept, this metabolic shift, it's happening. There is one research, I think from this year as well, which they shown that lactate can act on macrophage and drive metalloproteinase activation by macrophage and contribute in psoriasis. So I think it will be much more broad effect rather than just on gamma delta T cells.
Maybe just hooking up on that question, do you think it is Th17 specific? Because lactate can also upregulate interferon gamma, downregulates Th2. So do you think it's something which is rather specific for Th17 diseases, or does it have effects in, I don't know, other Th1 diseases?
So I think like when we look about like on lactate in general and T cells response, there are several papers in the context of cancer. So I think it's much more broad than just type 17 immunity, and I think it will be probably context dependent. But yeah, I think it's much more broad rather than just type 17, because most cells they have lactate receptors, lactate transporter receptors. So I think it will be much more broad.
Maybe you could allow me a brief follow-up question on that, because I think it's super interesting. I mean, lactate is something very general, as you mentioned. And my question is, if this increase in lactate levels, is this very local in the skin, or do you see also an increase in the serum, like systemically? And also, like when you treat psoriasis patients, do you see a decrease in the lactate? And then a third, which is also like related to this, if you do intensive training, your lactate levels increase. Does that affect psoriasis?
Yes, that's fantastic question. Yes, I think it's this is like the new concept, and I think it's very fascinating to really understand how metabolites, contribute to skin pathology. So with lactate, yes, there is increase of lactate in serum in patients. This paper from 2016, they measured the lactate level in the serum. So there is definitely increase in serum. Whether it's local in the skin, just like specific areas, we don't know. But I think what is happening, we think that keratinocytes, when they hyperproliferate, they need energy, so they relied on glycolysis, so and glucose. So there is a limited source of glucose probably in dermis. So that's why there is engagement of lactate in metabolism, for example, in T cells. The big question is whether it's involved in TCA cycle, or it's directly metabolites and act as a signaling. I think it's a very open question.
So what about the training?
Go ahead, Enikö.
Yeah. Well, unfortunately for this, I don't have direct answer, but I think it's would be quite interesting to see how this interfere with psoriasis development.
Thank you.
And patients.
Maybe a last follow-up question before moving to Camille. I mean, coming from Shruti's lab, do you think this circuit that you described is essential also in induction of epigenetic changes, because lactate plays a role in this epigenetic reprogramming and stuff like that? So you think that this is the way how in psoriasis and others epigenetic memory is actually developed?
Yeah, could be. We right now investigating memory in the context of psoriasis. I don't know if this is the direct lactate on the epigenetic level, but lactate is quite fascinating because it can act as a metabolite and act as a signaling molecule, for example, on post-translational modification like lactylation. So that would be quite interesting to see. Whether it's directly on epigenetic, I think it's a very open question, especially in the context of the memory, where I think it's quite interesting to see like how this looks in patients that result, for example, from psoriasis.
Thank you. Enikö, do you want to move for the next question for Camille?
So I think we received also a question from Patra Vijaykumar.
I'll hand it over. So it's again from Patra. So hi, Camille, nice presentation. Do you think these Staph aureus-specific TRMs would react to different strains of Staph aureus other than what you colonized on the skin if you challenge the skin? So if you take different strains of Staph aureus, and what about the reaction to different species in the same general genus like Staphylococcus epidermidis? So it's like 3 questions in one.
Thank you for this question. It's very interesting. The question of Vijay is, in fact, there's TRM are specific to Staph aureus, but in fact, are they specific to various factors, for instance, or to other proteins, other bacterial proteins? And that's a very good question. And for the moment, we don't have the answer. We check the specificity of T cells at the systemic levels. We did some lymphocyte proliferation test, for instance. And we demonstrated that we had a specificity of T cells restricted to only several virulence factor after application of E17, for example. We add some T cells specific to only 2 or 3 virulence factors and not other virulence factors, only suggesting that we have resident memory T cells which could react to only several virulence factors. And if we apply, for instance, other strains which do not produce.
And we lost him entirely.
Oh, we lost. Okay, I think that in that case, maybe he can answer this by email. So I think that in the interest of time, we have to conclude, and I would like to thank you very much, both of you, for your excellent presentations. Super interesting topics, much is happening. And I would also like to remind everyone who is listening that we have the next ESDR Kitchen episode in 1 month on the 18th of September, and this will be a talk given by Martin Bagor, identifying novel targets for the treatment of cutaneous T-cell lymphomas. Yes, and I would also like to remind you that if you missed some of these ESDR Kitchen episodes, they are all available at the YouTube channel of ESDR. You can see it here. And last but not least, importantly, in 2 weeks, I hope to see many of you at the 53rd annual ESDR meeting in Lisbon, Portugal. Do not miss it. Thank you very much.
Thanks a lot.
Thank you so much.
Thank you.
Bye-bye.